Hi, I'm Cassidy Kuchenbecker of Environmental Initiatives. We're going to talk about collection of samples for sewage. Before that, for anyone who doesn't know who I am or hasn't worked with me, I'm an indoor environmental consultant. I've been doing this work since 2003. I've done over 10,000 assessments for water damage, moisture issues, allergens, sewage, smoke, air quality. My educational background is in microbiology, chemistry, and immunology. I have both a bachelor's and a master's degree in that. I also function as a laboratory and I analyze all my own samples as well. So I'm going to have this discussion both as a consultant and from the laboratory side of things. So when it comes down to sewage, we have a much larger presentation than this, where we talk about various topics like health. When it comes to health and sewage, people usually think that we care about the sewage bacteria, like E. coli, and we actually don't. It's not the sewage bacteria that we worry about with health in a situation. You essentially would have to lick the floor of the sewage loss in order to get enough of these types of bacteria to actually cause an infection in our bodies. When it comes down to health and sewage, we care far more about the viruses that may be present because we need far less of those viruses to cause an infection. And we somewhat care about something called endotoxin, which is from gram-negative bacteria, but that's a whole different discussion. And then we also worry about mold if the sewage lasted long enough or the surfaces stayed wet long enough. But the sewage bacteria like E. coli themselves, that's not actually what gets you sick in sewage losses. So why do people think it's that? Well, it's because of what the laboratories test for. Testing for sewage bacteria is way easier than testing for anything else, and I'll explain that in a second. But let's first talk about bacteria and sewage as part of the whole idea of what is present in the environment. So first off, imagine there's millions of species of bacteria. Big group, big group. There's a smaller subset of that bacteria that we put into a category called coliform. Coliform bacteria have very similar nutrient needs and some other factors that put them into a good group together. Then, but those coliform bacteria are everywhere. They're in our dust, they're in our soils, they're all over the place. Then we have a more specific subset of coliform that we call fecal coliform. Fecal coliform are coliform bacteria that are far more associated with our intestines or with growing inside our bodies like that. And one genre or one genus of fecal coliforms is Escherichia E. coli. And there's many strains of E. coli. So E. coli, extremely specific for our intestines, although you will find it in the soils. It's just far more abundant in our intestines. Then fecal coliforms, also good to test for, but no one does, and I'll explain why. Because it's also associated with our intestines. And then coliform in general, which are very not specific to sewage, but there's a reason why we test for it. And then all bacteria. Okay, so normally when a person's going to take samples for sewage bacteria, they're going to be using a sterile swab and sterile water. They'll wet the swab and they'll swab the surface and send that swab to the laboratory. The laboratory will then extract the bacteria off the swab and put it onto a petri plate. Now, granted, there is DNA type of testing for sewage, which is pretty cool, but we're not covering that here. We're talking about the culture methods. So they then extract the bacteria, put it onto the petri plate, and then over a couple of days, the bacteria colonies will grow. Now, we use a very specific type of agar. The type of agar that we use changes colors based on some chemical changes that occur. First off, the coliform bacteria. Their colonies will eat the agar, cause a chemical reaction to happen that turns their colonies red. And then E. coli specifically, they will cause a chemical reaction that changes their colony color to blue, blue and red. Now, this sort of agar was actually developed for the food industry because they want to make sure everything is super clean. And so they care more about coliforms than we do for sewage. And it's a little bit more general of a test. So laboratories sell the idea of testing coliforms to you guys and to us, not because it's a good idea, but because that's the agar that they have available. But let me show you two plates to illustrate a point here. So first off, here is a what's called a contact plate, and I'll show you what that is in a second. It's a small petri plate, and you'll see a whole bunch of clear or yellow colonies. Those are typical bacteria. That's how bacteria look. They look clear, yellow, mucus, yucky looking, yellowish, you know. Then you see a few colonies that are red. Those would be the coliform colonies. And then you see a couple of colonies that are blue. That is E. coli. Now, so now we can see the different colors that show up on this plate. Here's another plate. This one is from someone's house. It's from the back of their television, I think, if I remember correctly. And you see a whole bunch of red colonies and a couple of blue. This is not sewage. This is not sewage. If you got this result back from a laboratory, they would say it was sewage because they had all these coliforms. But it's not. Those coliforms built up in that dust over time because they come in from the outdoors. They come in from the soils. And then, of course, you're going to have a couple E. coli here and there because we actually all have E. coli in our dust in our houses because we flush toilets. And so that's actually a pretty normal looking old dust test. Here's another contact plate, and this is directly from sewage. It looks completely different. Yes, you have some red colonies, and that's fine. You're going to, but then you see all these dark blue colonies. Lots and lots more, and that's E. coli. That is a true directly from sewage plate. So that's really what we're looking for. So what are these contact plates? We make these in our own lab, and anyone could. I think we're the only ones that do make them, but I love them. So they're small, they're yay big, right? You're gonna get them from me, if you do get them from me, and they're gonna be wrapped in, let's see if I have one around me, they're gonna be wrapped in a little piece of wax. And that helps so it doesn't dry out. So you'll just take this little wax off. Then you're going to sample a single surface. So I'm going to show you, you take the cap off like this. You take that, and let's say I'm just sample my palm here. You push it down on your palm, or on the surface, you push it on the surface, tap, 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 not too hard, tap, tap, tap. Turn it 90 degrees, tap it again in the same spot. You want to know how many colonies there are per square inch, so you can't just go all over the place with it. So the same spot. And you're going to put the top back on. You're going to then take tape. I don't care what you take. Any sort of tape. You're going to tape it on both sides like I'm doing here. Just a little piece of tape on each side just so it doesn't flop open. All right, now it's locked down. And then you're going to simply write on it with a pen. Let's call this one palm sample. You're just going to write on it your location so you know where it's from. Now, technically, you can analyze this yourself. In a few days, the colonies will grow. Actually, the colonies will grow within 24 hours. Usually, I wait 2 days for the color to really form before I start to count. If you send them to us, we'll send you a lab report back. If you do them internally, you can use them to test your own work to see how good your crew is doing. Or if you want to know if something was sewage, at the start of a project. You can, by the way, take swab samples and send them to us. And we'll do the same thing. We'll extract the particle, or we'll extract the bacteria off them and put them on petri plates, and then again tell you what the concentration of sewage bacteria are. We'll tell you total bacteria, we'll tell you coliform, and we'll tell you E. coli. Now, we do care about how much coliform and total bacteria are present afterwards, because what if you did your cleaning but there's a bunch of bacteria left? It means you didn't do a good job. So we're looking for low numbers of everything after all the work is done. Now, the question you might have is how many samples should you take? Well, if the idea is that you're trying to figure out if a water release was a sewage release, you only need a few, maybe 3, from 3 different spots where it is actually wet. Although, if you want to be very specific, you would do 3 spots from where it's wet, and you would do 3 spots that weren't impacted. So that you have a comparison between before and after the water impact. And if you're going to do like a post testing, like afterwards, let's say a 1,000 square foot basement had 2 inches of water. How many samples would you want to collect afterwards after you clean that floor? You'd probably collect 6 to 10. Multiple on the floors, and then a few on like the foundation wall, where the water came up some, and then maybe on some of the contents that got cleaned as well. Relying on 1 or 2 samples, it's often not good enough because you can have a really good spot here that's cleaned and a spot over there that wasn't quite as cleaned or wasn't quite as sanitized as well. So, 1,000 square foot basement, consider 6 to 10. And if you're doing a testing of these surfaces that have been impacted by sewage or by water, and you're not sure if it's actually sewage, you really only need a couple. You can get away with one. I would suggest three, just to make sure the results are correlating or close to each other. What you want to rely on is the E. coli concentration. The coliform concentration could be anything. It could be long buildup of dust, um, so don't rely on that number. You can do DNA testing. Like I said, there's those options that other labs provide. That's pretty cool. It's pricey. It's pricier than having a bunch of these contact plates, which are rather cheap. Other than that, if you have more questions, please give me a call. Otherwise, I'll be available for any phone or any emails as well. Thank you.